Our third speaker is Dr. Andres Carrasco, Professor of Molecular Embryology at the University of Buenos Aires. For many in my generation, our first taste of activism in biotechnology was the introduction of plants that were genetically engineered, I think by Monsanto, to resist the toxicity of glyphosate, which allowed more of that herbicide to be used than ever before. And then in June last year, Earth Open Source published a report by a group of international scientists called Roundup and Birth Defects. Is the public being kept in the dark? This document examined all of the original approval procedures for glyphosate and found that industry's own studies from as long ago as the 1980s, including some that were commissioned by Monsanto, showed that glyphosate causes birth defects, not only in laboratory animals, especially rabbits and rats, but now we're looking at cross-species effects. Dr. Carrasco will talk to us this morning about his excursions with glyphosate and the case studies of disease. Please join me to welcome Dr. Andres Carrasco to the podium.
Thank you very much for the invitation. Thank you, Bruce, and thank you, John. This morning, when I was taking my morning shower, I was asking myself why am I here? Because I am not a toxicology, I am not an expert in agrochemical or agrotoxics. I just spent my scientific career trying to understand the embryonic development using frogs and sometimes using chicken also. And during my career, I tried to always formulate the question, why? I understand that from the 17th century, there is a tension in science asking why versus how. How is what, in general, the things happen. But the why is getting loose in science. This is a kind of epistemological dilemma that probably is one of the answers what we are here today. Why men are using these chemicals and not how. A why that give you a feeling of the holistic view of nature, and the how in general conducted to technology and application, global market, money. Also, I would like to say that the answer to my question is this. It's very easy to see here that in 1996, the time that the GMO technological package was introduced in Argentina, the use of glyphosate grows to, and this is only to 2007, today is 200 million, 220 million liters per year, using the glyphosate used only in Argentina. You had to add this, what is used in Paraguay, and what is the amount that is used in Brazil and Uruguay. It's this territory, Argentina, Brazil, Paraguay, and Uruguay, constitute what Syngenta years ago called the Soy Republic. And we are using at the moment this increment of glyphosate for 15 years already, start in the middle of the 90s, in the 90s. This increment that, in my opinion, is caused by the GMO introduction, which is probably the first step in this kind of saga. Then Also, I would like to say that the glyphosate is a consequence of one particular definition, political definition, what kind of technology we are using for agriculture. And the GMO is the thing that we have to look now. This takes me to the point why I did these things. I was using frogs, I was using sometimes I was using chicken in order to understand embryonic development. But at the same time, I was traveling across the country, and I started to see populations, a small village where the people were saying that there was increase of cancer, increase of malformation. At the very beginning, that means something like 2005, 2006, was a very little discussion about these things in Argentina. But soon there were groups of environmental groups that would start to denounce, and these things got to the media. And then one day I decided that my lab could make a contribution trying to explain, not to verify what the people were saying in the field. People that were surrounded by soy plantations, people that were suffering fumigation on the village by plane or by mosquitoes. This is a machine that the people use to do the aspersion. Then there is no other motivation that what people was claiming for the scientists or for people that can give us some explanation what they are observing. And I say that because I believe that we have to take these opinions came from the society, when the society started to manifest some demands. And then we are talking about people, not about science today. At least in my opinion, science is something that tries to explain what is going on in society. When there is a market, there is an industry that uses this type of things. In my opinion, it could be that other ways to make agriculture, and not necessarily this way. And then it's something that the human being is provoking, and it's something very special and very delicate. As was done, was said, there were two documents, one from the National Research Council of Argentina, and another one from the University Litoral in Argentina, that claim that the glyphosate has nothing to do with any kind of harm in human health. On the contrary, these two studies produced by the group in England, they say they have collected information say the contrary. This is, you can imagine National Research Council and University of Litoral denying any kind of effect of glyphosate, based in the bibliographic revisions. Recently, we put all together, this is a group formed by 7 different groups and 7 different universities in Argentina that got together to make a kind of review what we are seeing in our country in terms of evidences in different science, ecology. We are seeing people working in the field with humans, people working in the field with frogs, ourselves working in the lab, with different disciplines, got together and put all the information that we have in Argentina concerning the glyphosate and some other agrochemicals. This is in press, and I expect to see soon. In 2010, we published a paper The paper was a glyphosate-based herbicide produced teratogenic effect on vertebrates and by impairing retinoic acid signaling. This is a fun story because at the very beginning, when I started with this experiment, I started with this type of experiment. Embryos cultured in the normal culture where you grow Xenopus levis, and now already know everyone what is Xenopus levis. Thank you, Tyler. It saved a lot of time. And put in the embryos in 1 and 5,000 dilution of the commercial brand, which means something like 430 micromolar. This is the first type of experiment. And then we start to observe clearly there were some malformations in these conditions. But the trick was, because we have the access to the technology, to try the second experiment, which was later on, where we inject, actually, I injected. After many years, I put it again in the mice setup, I inject embryos. I injected 200 to 300 picograms of glyphosate, what means in this volume, when the final concentration was 10 micromolar. But these experiments, both experiments, are different. One thing is to put the embryos and culture and grow the embryos in a flask with a saline solution, adding the glyphosate herbicide. And the other thing is inject a strict and very quantity of glyphosate equivalent drug inside the embryo. This second experiment was very criticized because the industry say that this doesn't occur in nature. There is a contradiction, a logical and epistemological contradiction. There is no way to have an effect if the glyphosate don't get inside the cell. Any cell, no? And then we inject a minimal amount of glyphosate in the cell to see what, in an embryo, 2-cell embryos, in order to see what happened during development. Because we spend most of our life looking at phenotypes. I'm sorry that the picture is not very clear here, but here you have a control with 2 markers. OTX, which is a marker that mark the brain, some regions in the brain of the animal. And Sonny Hedgehog is a gene that is expressed along the trunk from the tail to the head. And SOX9 is something similar, but also mark neural crest, which is a population of cells that are going to contribute with the formation of the head and the cranium. When you treated these embryos, you can see clearly that there is a change of size in the head. You lose a signal with the OTX and Sonny Hedgehog. There is a shortening of the tail, and the embryo is shorter. And also, because Sonny Hedgehog is a marker for the cardiac region, it was here, when you treat the embryo, this mesoderm, which is a cardiac mesoderm, disappears. In SOX9, it's happening exactly the same. You have a very similar phenotype, but labeled or revealed by a different marker. I was surprised, actually, when the glyphosate was injected in 1 or 2 cells. When the glyphosate was injected in 1 or 2 cells in 1 embryo of 2 cells, like it's here, one take the embryo into cell. You know, we know exactly what is the left and the right in the embryo. We can inject an amount of volume with the glyphosate diluted in one cell or in two cells. Then this embryo was injected in two cells, and what you get is, again, when using these two markers, is very similar to this, which was treated. There's a shortening of the embryo. There is a kind of a reduction of the cephalic region. There is a reduction of OTX label. Then, if you want to figure out how to call these malformations, you should say, that, for instance, there is a microcephaly. It's a small head. Microphthalmia, small eyes. Craniofacial disruption, because you have a deformation that we are going to see later on. Cardiac mesoderm reduced, like I say here, in a shortening of the anterior-posterior axis. Then all these malformations were observed at this stage, but if you go later stages and inject, for instance, this is a control embryo. You have two eyes. This is the head. This is the region, the abdominal region, and this is the tail. If you incubate this embryo until this stage, you get all this kind of alteration in this blue stuff, which are the cartilage, the cranial cartilage. But if you inject the embryos in one cell, what you get is something like that. You get one side, which is all the cartilage is deformed and have a shape that loses the correct shape, and the other side remain normally. You can compare this side with this side. This is non-injected side, this side, and this is the injected side. This is something that the frog allows us. When you have an embryo with two cells, you can inject one, and then you know that the one left or the right, doesn't matter which one, is going to be affected, and the other side is not going to be affected. The part of the panel, you have a control. You have an embryo injected in both sides, in one side. You have a normal side and the affected side. And you have the embryo injected in both sides. This is very important because this embryo has both eyes are in the front. When the two eyes are in the middle and fusion in the middle of the embryo, this is cyclopy. And this is a very well-known syndrome called, together with other malformations, called holoprosencephaly in human beings, in any vertebrate. Again, you have a microcephaly, microphthalmia, craniofacial cartilage shortening, and cyclopy. What I have to say that there is not difference in general terms. There is a difference of penetrance, probably when comparing the embryos treated with 1 in 5,000 compared with the embryos injected. But the type, the quality of the malformation is the same. This put very nervous everyone because it was not the adjuvants what caused any effect, was the glyphosate itself. And that is what these results suggest. So far, what we have is a malformation. It's the same type of malformation in two different experimental conditions. The problem for the embryologists is that you never can publish a paper if you have a malformation without to say more or less what is the cause. Those malformations, because we were working for many years with the development of the head in relation with different type of genes, bringing to the old experiments when we were using incubations with derivative of the vitamin A called retinoic acid. The retinoic acid is a very old friend of us. It's a very old friend of Bruce, me, and many others in embryology because the retinoic acid concentrations in the embryo activate and inactivate genes in places where the gene has to be activated or inactivated. It's what we call a morphogen. And then the distribution, the asymmetric distribution along the embryo of this morphogen, which is, again, it's a derivative of vitamin A, is in charge to activate genes in the correct places and the correct time. Then it's a kind of director that activate the genome in a very precise way. Those are, as I told you before, those malformations resemble the malformation that we already had obtained with retinoic acid. And then we decide to try to see how was the retinoic acid in the treated embryo. And then it took quite a while because we have to use a construct that activated is prepared when it's present of retinoic acid. This construct expresses a protein which is luminescent. You can measure luminescence in the tissue and in the struct of the embryo, and then we come out with this histogram that you have uninjected embryos. You have untreated embryos. There is a minimal amount of retinoic acid activity. This technique doesn't measure amount of retinoic acid in terms of molecules, measure activity of retinoic acid pathway. When we incubate the embryo with the herbicide, 1 in 3,000, we got an increment of retinoic acid activity. And measured by luminescence, right? When we reduce to 4,000, the activation of the activity reduces, and when we went to 1 in 5,000, it's almost normal. But remember that we start to observe the malformation in 1 in 5,000. That means that the morphology was more sensitive than the procedure that we were using as an indicator of activity. That is something that many people already said in this audience. And this is very interesting because you can see the effect without to be able to measure the cause of that effect. And, well, if you have an activity, increment of activity of retinoic acid, how do you prove that it's specific? But again, embryology, we learned that we can do rescues. And then we say, well, in the freezer, we have something that Bruce sent us 10 years ago. It's a blocking of a retinoic acid pathway. And then let's put the embryos. If we took 1 in 4,000, again, this bar, and co-incubate with this compound, which is a synthetic compound that blocks the retinoic acid pathway. And from here, we drop almost to the recover. I mean, what's a rescue? Reducing the amount of retinoic acid when you activity. But if you look the embryos, that'll give you a clear picture. If you have a control, you have an embryo treated, you treat it with the blocking also. It produces a malformation, of course. This compound block the retinoic acid pathway, and also you get the malformation. I mean, malformation related with retinoic acid is in both ways. You cannot have more than you need. You cannot have less than you need because it's a morphogen and it needs to be in very strict quantities in different parts of the embryo. Then if you increase, you produce, you increase the retinoic acid activity, you are going to have a malformation. If you decrease, also you're going to go to malformation. But if you put both together, glyphosate herbicide, which increase, withdraw, which block the retinoic, you recover the normal shape. And this is the final proof that these two embryos, that this, what we were observing here was specific. Glyphosate herbicide increased the retinoic acid activity and can be blocked by a specific, or more or less specific, general blocking of retinoic acid. That is in the histogram bioluminescence and also by phenotype. Well, what do we suppose? This is most of the experiment that we did at the time. We suppose that this retinoic acid alteration, it could be due to an increase of retinoic acid synthesis or a block of retinoic acid degradation. Actually, we know that the main regulator in the embryo is the degradation, not the synthesis. And then when you have this situation, you could alter, you can increase the retinoic acid in the embryo by these two steps in the enzymatic steps. But this increment of retinoic acid necessarily is going to affect the Hock genes, which is our genes that are expressing in the anterior-posterior axis of the embryo, the FGF, another gene, which when you knock out this gene, you have an anencephaly, you lose all the head, or the OTX, if you knock out by increment of retinoic acid, you have autocephaly. Okay? It's a syndrome where you lose all the mandible, and then you get some kind of holoprosencephalia or cyclopy. There are malformations in humans, even in animals. I have been observed an increment of malformation. I'm going to try to talk a bit about these things in the last part of my talk. And this malformation exists, but most of the women, the consult to the physician is abortion. They have abort one systematically. And then the real disease is not the malformation, it's loss of fertility for abortion, because the exposure to the agrochemicals, essentially 5 minutes. Ooh. I think Tyron have more time. Well, he talks so much faster. Okay, this is an illustration how the retinoic acid is distributed along the embryo. This is the head, and this is the tail. And then you have a concentration with gradients of retinoic acid. When we are disturbing with the glyphosate, are these things. As you can see, there is a large concentration of CYP26, which is the degradation enzyme in the head. The head cannot stand retinoic acid activity. It's release of any retinoic acid activity. Then the fur, which was going to be affected, is the head. In the same way that Tyron said that we are very similar in terms of hormones to humans, to frogs and humans, in terms of gene expression and control of morphogenesis, we also are very, very similar. Then I don't have to get in, but I would like to show these things because this is what is called caudal regression syndrome in mice caused by excess of retinoic acid. Essencephaly, microcephaly, microphthalmia, exophthalmia, holoprosencephalia, agnatia, microagnatia, gastrochesis. All these malformations, or some of them, in different combinations, appear in mice when you have an excess of retinoic acid. Then our hypothesis is that we are reproducing partially the malformation that already had been observed in clinics many years ago. When everyone knows that, when science, or physicians find out that the Accutane was a cream for avoid marks in abdominal marks post-pregnancy, was producing malformations, and the reason was because they have a retinoic acid, have a vitamin A in, and there was an increase of concentration. And then the retinoic acid, the vitamin A in excess, is a teratogen. A few other details, but I wanted to remark that there are two more agrochemicals, or agrotoxics, or insecticides. And here you have the papers which are associated, or these guys associated with retinoic acid metabolism. That takes me to the point that the retinoic acid is a very sensitive cascade. And anything that can block one of the enzymes, degradation enzymes, or the synthesis, or whatever, probably is going to cause this kind of malformation. That, suppose, if someone wants, this could be a test to see minimal amount of contamination, because the retinoic acid is not that the glyphosate is specific to block one enzymatic pathway. It's that the enzymatic pathway is really sensitive, and anything that can block an enzymatic dehydrogenation or oxidation, and what occurs from retinol to retinoic acid, is enough to give these effects. And that probably could be generalized for other agrochemicals, because it's the retinoic acid cascade, the secret, not the glyphosate. Many people say, well, how do you get the, well, this is in frog. You put the glyphosate inside the cell. Well, the glyphosate can access. What is happening in equivalent to man is happening in the frog. It's happening in 7 days to 10 days of gestation. Just when the embryo starts to be implanted in the endometrium, and all the vascular system is providing things to this small piece of cells. Then there is no placenta, but it would be placenta. The glyphosate also crossed the placenta in 15 to 20% from the blood concentration, have been published. Have been done experiments in rats by oral. Also, they found alteration of the shaping. Some people call variation of morphology. I call malformation. It's persistent in the soil. There are already data here in Argentina. They found 12 months later, they found still glyphosate or AMPA in the soil. There is no complete degradation. In my belief, it's not by ingesta, it's not by drinking, it's by breathing. The contamination is by inhalation, okay? The planes make this aerosol, and then people breathe these things. It's at least what they permanently denounce when they do the complaints. The retinoic acid has a half-life by intravenous 10 hours, but studies, toxicological studies and two-compartment studies, there is a concentration of glyphosate that is not eliminated from the body, that goes to the tissues, and then stay there. Okay. Thank you very much.
My question is what we know about the behavior of glyphosate in a pregnant mom and a human mother, and how might the chemical behave in the placenta And then what is its behavior in the fetus? Do we know how a fetus might be exposed? And do we also know anything about the half-life of the chemical in utero?
I have to say that more or less nothing, except in the crossed placenta and 50 or 20% concentration from the blood, and goes. This is our in vitro experiment. You take the placenta, fresh placenta, and then measure by perfusion how much glyphosate goes from one side to the other side. But there are no studies there are no studies in Argentina. There are no studies in blood because nobody wants to test glyphosate in blood. And about the tissues, concentration tissues, it will be interesting to know there is a bioaccumulation in different tissues. Of course, we don't have data of that. My presumption is that we are going to find in some tissues that we are going to have in the fetus for sure. Mm-hmm. I don't know if I answer your question, but we know very little about that because.